Let me see if I can get some of this out of the way. Yeah, I've always been interested in this metaphor of the Rosetta Stone, and it's a good metaphor in science because what it was parallel information. And that's a lot of what we do is we try to come up with parallel sets of information so we can verify what we're trying to measure in multiple ways. And I'll give some examples of that. So I'm going to talk a lot about, as Brian would say, applications of otoliths, and I'm going to concentrate on striped bass, but this is just a small representation of the amount of research that's been going on with this structure and its application to fisheries and management and ecology. But before I do that, I'm going to talk about what otoliths do for fishes. They weren't built for fishery biologists, so we should think a bit about what they do for fishes and how they function. All right, otolith, not the hemolith, not the gastrolith, but the otolith. So ear stone. And here it is. This is kind of a typical three otolith structure within the labyrinth system. With it, you see the semicircular canals there, and then you see these big kind of vesicles of epithelial tissue. That's the whitish tissue there, and they surround three otoliths, and there's two labyrinth structures, one on either side of the head. And the largest otolith is the one that I'm mostly going to be talking about. That's called the sagita. And these otoliths are located in the basal part of the neurocranium of fishes. So unfortunately, when you extract them, it's usually a lethal operation. There's a concavity in the neurocranium called the otic bulla, and they rest in there. And the otolith, you can see how it sits in that sac. It's in its own orbit. It's in thick endolymph, and it allows the otolith to kind of move independently of the fish, and that's very important for structure and function. So humans have otoliths, but we have lots of them, thousands, and they're called otoconia sometimes. Maybe you rotate your head, maybe you can feel the sands moving along your macula, and that's telling you something about angular acceleration. It's telling you about your equilibrium. And this is also important for humans. But in fish, otoliths are big monomorphic crystals, and they have very characteristic shapes for the species. They're much bigger than the otoliths in humans. The otoliths sit on a bed of cilia, of stereocilia structures. And you can see here, this is the dense otolith, and here are the cilia. And the cilia are located on a bed of neural tissue called the macula. It's not completely surrounding the otolith. It's just underlying part of the otolith. Now, the way I like to think about or try to describe how otoliths function is if you put up your hand and you held a can of Coke, or I have this glass of water, that's dangerous. But imagine if I take my other hand and I whack my forearm. My hand and finger is going to lag behind. That's called a shear, that differential movement. And that's what happens here, is that something whacks the fish moves suddenly, and the more dense otolith lags behind, and that shear force is translated to these cilial structures that create a neural impulse that goes to the brain. And the brain can then, through the complicated kind of different patterning of the macula and the cilia, it can discern the direction of that shear. And in terms of hearing, which is really amazing, they can determine the bearing and the amplitude of sound. Interestingly, sound received by the otolith is not in terms of sound pressure, but particle motion. Fish hear by particle motion. The sound makes water particles vibrate. And in the near field, within meters, that's what the otolith detects on this bed of cilia. So otoliths come in all kinds of shapes and sizes. In a species, otoliths will keep growing as the fish grows. But across species, we see all kinds of interesting differences in shapes and sizes. So on my finger here, this is an otolith from a 500-pound bluefin tuna. right? And these, which are earrings, are much larger than that, are from a red drum. And these are from a cod. So you can see that they all have different shapes. And what we think about the size, anyway, is that it has a lot to do with the hearing capability of the fish. So fish that are good hearers, fish that produce sound, soniferous fish like drums, red drum, black drum, toadfish, they tend to have big otoliths. And we think that helps them function hearing. Other fish with big otoliths or bigger otoliths, including cod and striped bass, we think it helps them with maneuvering, that angular acceleration. They're good maneuverers as compared to bluefin tuna, which is very good at going in a straight line very fast. Otoliths form early in life. This is a larval striped bass, just a few days old. You see its eye and its forming mouth and its yolk. You look carefully, you can even see the otoliths. They're in there. What I've done here is I've embedded the fish in plastic, and then I've sectioned through here, and this is the result. Here are otoliths in just a few day old striped bass larvae, and they are 10 micrometers across, so they're tiny. That's why I had to section the whole larva. So they're formed really from hatching in these fish. And it says that their function is important throughout life. It's important during the larval stage, juvenile stage, adult stage. Now onto the metaphor, and this is Champollion, and for centuries, 17th, 18th centuries, Egyptologists struggled with all these glyphs, hieroglyphs that they're unearthing in Egyptian ruins. They had no way of translating until they had the Rosetta stone. This is a stone that had parallel languages, the hieroglyphics on top, and ancient Egyptian, and then Coptic language below. And this suddenly allowed all these glyphs to be translated. And so it's a nice metaphor for any number of things, but because we're talking about stones, I thought Rosetta Stone worked well. And we're going to show you how we've translated otoliths as their own Rosetta Stones. There was really a transformational discovery with otoliths that was published in 1971, not by a fish biologist, but by a Yale geologist in science, Giorgio Pannella from Yale University. He published a paper, and he said, these little rings in the internal structure of otoliths that you guys have described over these years and kind of ignored, I think they're daily. These are daily rings. And we said, really? And he provided some evidence for that. And this was just a groundbreaker. We don't have structures, hard structures, I think, in any animals that provide this kind of daily resolution. And daily resolution becomes really important for fish because things happen very quickly early in their life, as I'll show. So that resulted in just kind of a revolution of applications in terms of fish otolith studies. Almost any species you can name that are members of the teleos, the bony fishes, you'll find otolith applications. We have tens of thousands of papers, thousands of scientists. We've had 7 international symposium proceedings. So it's really its own subdiscipline within fisheries ecology. One of the great things about otoliths, if you use careful technology, is that the calcium carbonate structure, so these are the same kind of structure you see in clamshells and so on, you can use metallurgical techniques, kind of techniques you would use on gemstones, pearls, and you can basically take a slice out of the middle of the otolith and expose these age structures. So this is kind of like an onion. Take a slice out of the onion, hold it up to the light. You can see the rings much more carefully, but here we're working at different observation scales. So if we want to understand larvae, we're looking at the most internal structure, and these individual daily rings are increment or microns across, micrometers across, very small scale. And then we can look at daily rings in juveniles at a bigger scale. And then still at a larger scale, we can look at yearly rings in adults. So you can see the structure can give us a whole range of information across the entire life cycle of the fish. Here are some of the techniques we use. We embed otoliths in plastic so that we can carefully section them with this metallurgical saw that has diamonds embedded in these wafer saws, wafer blades. We use careful microscopy. We use polishing compound, careful grinding, and we get a section that looks like this. This is the most internal part of the otolith. This is the earliest part of the fish's life. And then these numbers apply to different years of that fish's life, just like tree rings. Well, we had to go out first and confirm that Pannella was right. So there were initially many of us, including myself, went out and determined whether these rings, these little striations we were seeing inside otoliths, were in fact formed at a daily rate. And I kind of did something that was not very nice for the fish. I decided if they were forming at a daily rate, we should challenge this hypothesis, so I starved them. That's not a friendly thing to do, but it turns out striped bass larvae are very resilient. So I starved them for several weeks and I reared them. And you can see that even when you starve them, these are the rings, this 23-year-old striped bass. We could find the rings using careful electron microscopy. By the way, you can, I think, I need to get rid of this. Sorry. Oh. But you can see that in the red circle area, you can see little sparkling things. Those are otoliths, formed very early in life. So we're going to ask a question now. And this question I pursued when I first arrived at Chesapeake Biological Lab. It was prompted by kind of my hero, Romeo Mansuetti, who's one of our pioneering scientists at CBL and at your society. And 62 years ago, he and Hollis produced this map from their own sampling and other samplings that showed where striped bass spawned throughout the Chesapeake. Those are those stippled areas. And Mansuetti made this observation that these spawning areas, the lower boundary is the salt line, he called it. That's the interface between freshwater, tidal freshwater, and tidal brackish water. He said, it doesn't matter which system, the downstream the limit of where we get eggs and larvae, that's that salt line. And he left it that. And question was, well, why would striped bass adults that are in the Chesapeake or in ocean waters, why would they come all the way back up to this freshwater area to spawn and do this year after year? It must be good for their eggs and embryos and larvae. What happens to them after they're spawned? Here's an example of some embryos that are just hatched. So I looked at the system on the eastern shore, the Nanticoke River, to investigate this idea that the salt front is the delimiting line for nursery areas and what happens after spawning occurs. Besides the daily rings in otoliths, and here's a 30-day-old striped bass, you can see the daily rings. I had another trick up my sleeve. As part of my PhD, I figured out how to mark millions of larvae by dunking them in tetracycline, which leaves a mark on their otoliths. So I would immerse, say, 5-day-old striped bass larvae in these tetracycline baths, and I would mark them, and then I would grow them out. So here's the mark at 5 days, and this is the rest of the otolith at 30 days. And this is a kind of a permanent record. And I can mark them at different days. I can do multiple marks. So we can actually code larvae by marking them in this way, at least for hatchery fish. And then I had at my disposal, which was kind of great, a period in our striped bass management where the hatchery had been used, you can see that down here, in the 80s to prompt recovery of striped bass. It never really had much of a role. It was really relaxing the fishing pressure on them. But we wanted to hedge our bets, management, so they had a very large hatchery program. And I arrived in 91, just as striped bass were recovering, and they didn't need to do the stocking anymore. They didn't need to release all the juveniles year after year. And I said, hey, we got this great idea. And so they let me have access to their millions of larvae that they produce. And I mark them with the different chemical markers. And here we are in the hatchery. And that's me at a much younger age. And this over here, let's see, if you look up here, all these little wispy things, those are the larvae that we've marked. So there's 100,000 larvae just in this one container. And that tells you that this is why it's a feasible approach, that I don't have to mark a high volume of water to mark all this larvae. So what do we do next? We went to the Nanticoke River, went to a pier or a couple piers, different sites. And in 1992 and 93, each year we released 13 million striped bass larvae that had these chemical marks right into the drink. And then we had this kind of crazy thing like pickup sticks, says we're going to go find them again. So there's natural reproduction going on. There's wild larvae going in here as well. So we're going to have to filter a lot of wild larvae to find our marked larvae that have their otoliths marked. And it turned out they were about 5% of what was out there had our marks in them after we released them. So here I can see one. You can see I'm pointing to the mark in the water. There it is. Go over there. And so we use kind of quantitative plankton toes to get densities, which we then expanded to river-wide abundances. And here's some of the larvae that we caught in our nets. All right, first complicated slide. Gird yourself. So let me take you through this. Each one of these graphs depicts either wild or hatchery release larvae in either 92 or 93. So let's start in the middle with these black dots, and these give our estimated abundances in different parts of the river at different times. So for our mark larvae, remember we release 13 million in each year. The bigger dot is greater than 1 million, and then we go down to less than 100,000, and that corresponds to these top plots here. For the wild larvae, there's a lot more out there. You can see we start out at greater than 100 million for our bigger dots, and we go down to less than 100,000. So each one of these spots is a density or an abundance that we've estimated for this date and for this part of the river. The river is divided up between freshwater, which is blue, and brackish water. And we have finer scales here based on conductivity, which I call saltiness over here. And what you really want to follow is this light blue line. That's the salt front. So the salt front is moving up and down. And the reason for that is those are storms or cold fronts coming through during the spring season. We're going from April to May. And so we move upriver to these higher river kilometers, we're in freshwater. We move downriver, we're in the brackish water part of the estuary. And Mansuetti said, well, everything should be in freshwater, should be above that salt line. And you can see that's the case for our released hatchery larvae in 1992 and in 1993. And that's also true for our wild larvae. We don't see them below that salt front. So we confirm that part of the idea. The other thing you might notice is that the circles tend to be a lot bigger in 93 than 92. So 93 was a better year for larval survival and production than 92. And the final thing I want to point out here is there's a lot more blue in 93 than 92. So the salt front was displaced down estuary. So we would say that the larval nursery was much bigger in 93, 92. So the salt front is moving around year to year, depending on how wet the conditions are. So we had multiple batches, and that's what these dots are in this plot. And this is daily mortality. So we go from 18% daily mortality, 18% are dying per day of our marked larvae. And we're going down from 18 to about 5%. And you can see overall it relates to temperature. So you have warmer temperatures. If you have colder temperatures, we have mortality rates at about 15% per day. So that says that it's better to be warmer, but also that's the result of the effect of those storms coming through. They're the things that cool down the river. The spring temperatures are gradually warming, but you have the cold fronts and they temporarily depress those temperatures. One storm came in and was very extreme, And the release group here, we never saw again after we released it. That group is right here. So that was our most extreme storm. We released a group there, and they disappeared from us. They presumably all died. So we see that 93, the final result was this was spring, and eventually these larvae became juveniles. So we went out in the summer, we collected juveniles, and we looked for marks there as well, or otolith marks. So we found out the juveniles in 93, they had 4 times the number than we did in 92. So the improved survival that we saw in 93, the bigger nursery area we saw in 93 carried forward to having 4-fold the abundance of juveniles in 93. Well, let's put this together. And here's the overall idea, is that in 93, we had a much bigger nursery. The salt front was moved down. It was wetter. We had higher precipitation rates. And we're suggesting that that relates to buffering in the system. You have cold fronts coming through, which I represent here as a bucket of ice. If you pour the bucket of ice in a small nursery area, it's going to have a much bigger impact than if you throw that bucket of ice into a bigger nursery area. So we call this the nursery volume hypothesis, and this shows kind of the relative dimensions of the Nanticoke River nursery. You can see it. This was the salt front region here in 1992 at river kilometer 36. And in 93, it was all the way down by Vienna at river kilometer 32, a much bigger nursery area and better buffered against the storms that occur. The other thing is, how do these big nursery systems form? They don't form right when we're out there sampling. They're the result of late winter and spring weather conditions. If you have a lot of snow, if you have a lot of rain during that period, you're going to end up with a bigger nursery. So the weather conditions in late winter and spring really determine whether you're going to have a good year for larval and juvenile production. So in summary, otoliths have shown us a lot about how striped bass survive in their nursery environments. Salt front was definitely saw it was a delimiter in terms of where striped bass larvae occurred. They were retained above it, as we say in our oceanographic parlance. And through its influence on nursery volume, late winters and early spring precipitation precondition the system to either have a good or poor juvenile production. Nursery volume buffering against bad weather. All right, that's number 1. Number 2, we'll go back to Professor Mansueti, and he pioneered age and growth studies on striped bass, and he also had these wonderful illustrations. He was at the Society of wonderful illustrations of reptiles and amphibians, but he carried that forward. And, I was looking at this today, when it was just, these days in PowerPoint or whatever, you would just make one of these images and you would just copy, you just make it bigger. Each one of those images is individually drawn. It's just really great, the attention to detail. And it's a very nice demonstration for the layperson about how striped bass grow. The darker ones are males, the lighter ones are females. They used to use these nice Roman numerals for ages back then. This goes up to 14. That's our largest, biggest fish. Well, since then, there's been pretty good literature out there that says that scales which were used by Mansueti can be biased. So I wanted to look at scales, if they do an adequate job in aging striped bass, and maybe otoliths do a better job. So I went back to the hatchery program. And what the hatchery program did is released 1 to 2 million juveniles. These are young of the year, 3 to 5 month old juveniles each year during this hatchery program period. That was remarkable. They had a trailer set up with workers kind of working day and night, injecting juvenile striped bass with these little coated tags. I'm sure they injected a few of their fingers along the way too when they got tired. But in any case, they had all these tagged fish out there. They released them. And then various survey programs would detect them, and they would then take the scales and otoliths, and they let me have some. And so I could relate how many rings were in the otolith or the scale to how many years we knew that they were at large, because we knew from the tag when they had been released. And what I found was that otoliths did provide much more accurate ages than scales, particularly for older fish older than 10 years of age. So since then, otoliths have been kind of the preferred approach, although they've disadvantages. You have to kill the fish, get them. Scales, you don't have to. Well, at the same time, I was looking at a new fishery. This was called the trophy striped bass fishery. It was brand new in Maryland. It happened just after our fishing ban, our moratorium in the 90s. And this allowed fishermen kind of towards the end of the spawning season in May to catch the adults that were 36 inches or larger. So these are trophy fish, big fish, provide me an opportunity to look at age structure. And so one of the fish I first encountered was a fairly large fish. It was 20 kilograms, 45 pounds, over a meter in length. And I was kind of amazed. All those little arrows. So you could do this as well as I could. It's very clear. It's 31 years old fish. And I was like, wow, that's older than I am at the time. Older than that now. So it was kind of an epiphany for me. It's like, why wouldn't striped bass live so long? What's the point? You know, they can. So, and so I went a little bit far afield and decided to go hug a sequoia. But the sequoia is really the epitome of longevity and size, isn't it? And the reason that sequoias live as long as they do and get as big as they do is that they're oversampling. They're reproductively oversampling in an uncertain world. So in most years, seedlings from the sequoia amount to zilch. It's the rare event, often associated with fire events, that provide enough forest clearing that the seedling actually succeeds. So a sequoia lives 100 years because it might take 100 years for its seedling actually to find good enough habitat and replace the adult. And that's the same thing with striped bass. That's why they live so long. So that they can span decades of early survival conditions that are mostly not too great. It's only that odd year where they're pretty good. And then so they're spanning those periods of poor reproduction, take advantage of the odd year that actually is favorable for their young. So I got a picture of this in 92 when this is the entire trophy sample that I looked at, fish that were over 36 inches in 92. I hope you can see it's a little light. But let's start over here. You can see that I have in my sample of fish that are over 36 inches in 1992, a lot of fish that are 20 years or older. You see, I found actually a couple 31-year-olds. These fish are really old. And they were born in the 70s and 60s, really old. Why are they out there? Why hadn't they been fished? You know, it's a long time to be around if there's fishing out there. Well, it turns out that historically they were protected. We had a size limit. You couldn't catch fish over 36 inches in the Chesapeake Bay. This trophy season was new. Previously, people just wanted to catch pan-sized fish, which were 12, 15 inches. Those were the tasty fish that people wanted to bring home. So they were protected, and they actually stimulated this recovery that happened during this period of very low abundance for striped bass. You can see it's almost, you can think of it as a nick of time, because look at this. This is like a war hit this population. There are 10 years of absent ages. And this is a period of extreme overfishing. This is when recreational fishing really ramped up in the 70s. It's capturing those pan-sized fish, but also things like gill nets were becoming much more, I don't know, grossly deployed. It was a gauntlet in our tributaries, and they estimated for that time small fish were being exploited about 90% per year. And so it was overfishing. And then these younger big fish, they're the ones that are part of the recovery. They're coming in. And so this goes to the point that these old fish They're very important for striped bass recovery. But there's another part of this story too about why that's from a management point. It's great we had those old fish to stimulate, but why might they occur in nature? And the reason for that is that we have these droughts of reproduction, droughts of periods where we're having poor juvenile production. This is juvenile production measured by a SANE index by Maryland Department of Natural Resources. And I've circled this period when it was really low in the mid 80s, a 6-year period between 82 and 89. And this is when they were stocking fish. They had a fishing ban. They did all kinds of things. They called it a striped bass emergency to try to get the striped bass spawning stock up and to get them reproducing again. And you can see that worked. And this one, this is our old females. I was in 92 sampling them, but these old females, they were the ones that really contributed to those recovery years of juveniles. Now, I have to say that we're looking at another emergency. This now is 7 years. So remarkably, in our own times, we're in uncharted waters. Recruitment has never been so low. And you'll be reading about this in the paper in the coming days. This number is just out. So what does striped bass longevity do? It is bookends these periods of very low juvenile production. We spawn year after year after year because we have a low batting average. So longevity in otoliths, we certainly have shown that otoliths have give us new insights into longevity for striped bass and other species. Striped bass is one of our oldest species in the bay, with longevities exceeding 30 years. Striped bass live a long time because their reproductive batting average is low. They need lots of time at bat. And management agencies now recognize this and use age structure and longevity as a key indicator of sustainability. All right, the last bit has to do with migration, of course. Can't leave that alone. So I've talked about how the nursery up in our freshwater population.
Dave, break it up on us a little bit.
Relates to longevity. It's poor. Okay. Can you hear me okay now?
Yeah.
Okay. So this is an uncertain kind of event on an annual basis, so that contributes to longevity of striped bass. But the other thing that striped bass do that we know is they migrate. And striped bass that migrate from the Chesapeake are the most important in terms of contributing to coastal U.S. state coastal fisheries. So if you're in Massachusetts, if you're in New York, most of those fish you're catching in shelf waters originated in the Chesapeake Bay. So it's very important from a management point of view to understand why or how striped bass, what size, how many striped bass leave the Chesapeake Bay. And here we go back to our hero, Romeo Mansuetti. He did this wonderful tagging study. I can't imagine, it must have been so ambitious because you can tag, you're looking at ocean migration. So this is vast area where you're having to try to get tags from. So he tags fish with recognizable external tags, Peterson tag, nylon tag, ring tag, and he sets them out. And in the Chesapeake gets lots of recaptures, and that's shown in this little inset. And the recaptures show that as a fish gets bigger, they tend to migrate farther. That's just within the Chesapeake. Getting recaptures outside the Chesapeake is hard. You know, people don't know you're doing this. There's no advertising, there's no telecommunications in the 60s, but he has some network of scientists and he gets a handful of recoveries back. And this is a wonderful diagram that shows the map, it shows the coastal area, and it shows these functions of size versus migration. And here the size versus migration function on this dashed line at 30 inches right down here. We have about half of these recaptures in coastal areas coming back or being shown. So 30 inches is kind of a threshold size where fish are expected to start migrating into coastal waters. It says after 30 inches, most of them are moving into coastal waters. He wasn't very confident about that because he had so few recaptures from coastal areas. And so this provided an opportunity for me to look at this from a different vantage. So now we're going to go back to our otoliths. We're going to do a little bit deeper dive here. So here is our otolith with the annual rings, the yearly rings. And here's the first life. And I kind of made a inset of that. This is the larval period right in here. You can see some of the daily rings. And I've used what's called an electron microprobe to get from the larval period all throughout this first year of life. This is the first yearly ring right there. And then you can keep going. Each one of these spots is an electron that's bombarding the sample. It causes X-rays to be emitted. The X-rays have characteristics of certain elements or isotopes. In this case, we're going to be looking for strontium. Each one of these spots is about 5 micrometers across. You can see they're spaced very tightly, so we're getting very good resolution by this approach. And by this approach, because we're going from the first year of life all the way out to the end, we're basically making a time series of the fish's life and saying this is how strontium varies across all these ages. And we're looking for different patterns. Maybe it goes up, maybe it goes straight. But why would we be looking at strontium? Well, it turns out that strontium relates to salinity in this way in many systems. It's proportional to salinity. It represents salinity. It dilutes, just like salinity does, between freshwater, between marine water and freshwater. And so we can use strontium as an indicator of salinity. And striped bass migrate between different salinity zones. As Mansuetti and I showed in our study, they're going to go up into fresh water to spawn. They're going to do that every year. They're going to start life in fresh water. They're going to spend some time in brackish, mid-salinity waters, and then eventually maybe they're going to go out in full-strength salinities. So we measure strontium across this 27-year-old individual. Each one of these are those annual yearly rings. And what we can see here, this is the result for strontium. It's low strontium earlier in life. It gets higher later in life. So this might indicate Chesapeake existence for the first 8 or 9 or 10 years, and then it moves into ocean waters. And the up and down, maybe you can figure what that is. Those are the spawning migrations. Year after year, it's moving into fresh water. We're getting an indicator of that, going down to an nadir each year and then coming back up into ocean water. So we looked at annual averages or age-related averages. And let me start here. This is a 2-dimensional representation of strontium in the otolith. And here, this just shows very nicely the red is high strontium, the green is low strontium. You can see the banding. So this is those spawning migrations. We looked at average age strontium levels and from that inferred salinity. And for males, 4 to 6 years of age, about 40%, about 20% left and went into the ocean waters. So a small proportion. And then we start getting to about close to 50% for males and females in 7 to 9-year-old category. But then after 9, we see that the majority do tend to go into ocean environments. So a 9-year-old striped bass is approximately 30 inches. And this aligns with Mansuetti's work. So we've confirmed it with a different kind of approach. But this is not the only thing we saw. Not all big striped bass or old striped bass migrate. Some stay their whole life in freshwater. Others spend their whole life in the estuary, as shown here. So we have mid-salinities, and then others are oceanic. So we have different migration types within the population. And maybe this reminds you of something. Our geese, birds do this. So why wouldn't fish do this? This is called partial migration. Some are resident, some are migratory, or in striped bass, maybe there's multiple different migration types. Some are estuarine and others oceanic. So partial migration in my research and exploration literature, it has been increasingly common in fishes. mostly understood now through these otolith chemistry applications. And this really drove my book on migration ecology and marine fishes, which is kind of a parallel treatment of birds and fishes. There's a lot of comparative ecology between what we know about birds, because bird migration, I hate to say it, but birds are a lot easier to track around than fish. But the theory is a little bit more developed for birds than fish, so I borrow heavily from it. All right, wrapping up. Since Pannella, otolith science has led to the translation of microstructural and microchemical features to seminal life history information for the teleos. This is not so easily applied to things like sharks, sturgeon, and so on. It's transformed our understanding on the previously hidden lives of fishes. We're integrating temporal and spatial scales across the entire life cycle. So otolith science is taking us into small streams and rivers and estuaries and bays and entire ocean basins. And also we're looking at larvae and juveniles and adults. So it's really a very nice tool for integrating across large dimension scales. And then finally, it's contributed to improved fisheries assessment and management. I thank you for your attention. Happy to take any questions.
Well, thank you. That was amazing. We'll have you come back and unshare, and I'll put a spotlight on you.
Okay.
All right, let me find a spotlight. All right, and we're going to open it up for questions. You can raise your hand. You can put a question in the chat box. If you raise your hand, I'll let you unmute and ask one. Sarah already has a question in here. When the number of fish reproducing becomes low during the drought periods, is there any concern about low genetic diversity in the preceding populations?
I haven't heard any. I mean, the abundances of striped bass are still pretty high. So things like effective population size and breeding haven't typically been a consideration, but we have had situations historically where that may have happened. And that's certainly happened over glacial periods, because these fish, they colonize and then they become extirpated as with waxing and waning glaciers, or certainly survived glacial epochs. I wonder about like the Delaware. It was kind of surprised, but the Delaware was just awful in terms of water quality. And this kind of tidal freshwater region where striped bass spawn and that larvae occur, it was hypoxic, anoxic, polluted, and a lot of people wondered if things like shad and striped bass still lived there. They did, turned out, but that kind of population collapse, maybe there's a genetic signal there. It'd be something that you might actually be able to find in the literature, because they've done a lot of genetics work with striped bass.
All right, Rachel asks, what would sampling or analyzing otoliths look like for early career fisheries tech?
What would it look like for early career fisheries technician?
Yeah, for an early career fisheries tech, yeah.
Yeah, there are age and growth labs that are federal and state and private. So that is kind of a career trajectory. And then a lot of researchers, as I said, use them. I worked early in my career as a grad student. My co-author was a fisheries tech, and she was like the supreme otolith preparer. She could just, it's a little bit of a Zen thing with making these otoliths, that you are trying to find, especially for the daily ring stuff, you're trying to find an area that's like 10 micrometers as you're polishing down. And if you go past, it's like close but no cigar. So it does take a lot of practice and patience and a little bit of Zen for some of these kind. But for the adult otoliths, it's pretty straightforward. They have a very large lab up at Woods Hole. They have a very large federal lab at Panama City. And so if that's something that interests you, maybe I can help make connections. I'm trying to think. DNR does not have an aging lab. The other states do.
So are there any do people or researchers collect otoliths from fish markets as well to get different data, and are they studying it that way?
They sure do. And yeah, I've done a lot of work on bluefin tuna, and I've made a real nuisance of myself in some fish markets, going to Madrid and knowing enough Spanish to ask to purchase young bluefin tuna in the market. And then, of course, you're left with the dilemma of where are you going to take the otoliths out? Downtown Madrid. But yeah, there's any number of ways that you can collect fish. And the striped bass that I've looked at, try to use fishery samples so I don't have to go and kill them myself.
I was going to ask, let me ask one more question before I turn it over. It's, we had on here Adrian before, and he's into fossils, and he said that otoliths are a fossil that is collected. And are you able, do you know anything about fossilized otoliths? And are they able to either measure the strontium in those at that point? I know that you're able to measure the different isotopes in kind of woolly mammoth teeth and things like that.
Yeah.
I didn't know if there was anything similar.
Yeah, Pannella's paper in Science, that one I featured, he used some fossil otoliths in that paper as well, because he was interested in paleontological applications. And I remember I've looked at them, and I couldn't believe that you get information from them, but you can. You can get age information, and you can get isotopic information. They've used it to reconstruct like droughts. They find otoliths in lake beds of Australia, and they can reconstruct droughts across millennia. So yeah, it's definitely has an application there.
All right, Alan has his hand up. Go ahead, Alan.
Hi, David. Thanks so much. So interesting. Two parts or two questions. First one, I don't know if I missed how it was decided that we know that those are daily rings. I think I saw the delineation of sort of what shows like an annual ring, but so I just how did someone figure out they were daily, number one, and number two, what causes that daily ring? Is it rotational? Is it day, night, light, dark? And then my second question is my standard question here, and you're going to get the same treatment as everybody else that speaks here, which is what got you interested in this? When or in your life as a child? And then sort of how did it then become this specific area too? So take them in any order you'd like.
Yeah, that's a great question. So the easiest way to get the daily stuff is we can rear fish. That's why I'm hanging around hatcheries all the time, because I can get the fish, I can rear them, I can grow them out known age, and sample them periodically and just relate their known age to what I see in the otoliths. There's other ways you can do it. You could release mark larvae like I did and then collect them in the field. I did pond rearing studies to better emulate wild conditions. So there is a very large literature in relating known age fish that you've reared or somehow you know that what their age is to what you see. The dial, the daily increment part is related to how these otoliths are they're organic matrices, and the organic stuff is actually deposited from that epithelial tissue that surrounds it, and it creates this matrix. And there's a dial rhythm in terms of that matrix, that proteinaceous matrix. There's some kind of circadian rhythm associated with its formation, and that causes zones to be more highly calcified or to be increasingly organic. And that creates that optical pattern that we see as daily increments. That's the kind of current understanding. Why I got into this was, well, a couple of things. One, of course, Jacques Cousteau. When I was a kid, that was The Secret Life of Fish, and Rachel Carson and the rest. I give them due credit in my book. But the other thing is I had a left turn as a grad student. I really was interested in aquaculture. So I went to Japan and studied aquaculture, and I kind of hated it. It's too hard. I was killing so many fish. If you do something wrong, if the fish die, it's your fault. But in nature, it's different. And I was always fascinated with this idea that you don't see what's going on below this opaque surface. I think that's really the whole Jacques Cousteau thing as well. So the otoliths really help you have an indirect way of understanding these hidden lives of fish.
Thanks so much. Actually, brief follow-up question. Have they ever looked at the otoliths in humans to look at the same kind of rings or aging or anything?
I don't think it's possible. I don't know, but the gastroliths they've used and the statoliths they've used. Statoliths are in the cephalopods, and they've used those for aging. They get daily rings in those. And the gastroliths are a little bit more difficult. Those are in things like crustaceans that try to use those for aging purposes as well. But
And
yeah, I
I
don't
also,
think you could use otoliths. And I have tried to use otoliths in other fishes like sturgeon and coelacanths that don't have these nice monolithic calcitic ragonitic structures that the teleos do, and it doesn't turn out too well.
And with humans, I wasn't talking about like killing a human. I was talking like, like an autopsy
Oh yeah,
and knowing the person's 79 years old and then looking at the otolith in their ear.
yeah, but the otoliths in humans, there are thousands of them. They're just like little grains of sand, so I wouldn't know what to do with them.
Alan, that's when your crystals get aligned and you get the vertigo kind of thing. That's your
No,
otolith.
yeah, I knew that because I had vertigo and I remember her talking about the crystals, the physical therapist. So yeah, interesting.
Really? I don't know about that. That's interesting. So
Yeah,
that's a
they'll
Yeah, yeah, they have you do this procedure where you're sitting up and they have you sort of fall over to the side really quickly to try to sort of realign the crystals to move them around. And they also do it to test whether that's the cause of the vertigo.
oh, I didn't know you could do that.
Timothy asks, is strontium in otoliths present as a carbonate?
Yes, it substitutes in for the calcium. It's a little bit bigger, but it fits in nicely. It tends to substitute in a temperature specific way. So it says that it's kind of kinetics that causes it to substitute in. It's in parts per thousand. Otoliths are really pure. They're like 95% calcium carbonate. And strontium is one of the higher impurities within otoliths.
Steve asks, what is known about otolith size, morphology, and chemistry of deep sea fish in comparison to shallower level fish?
I couldn't tell you. I mean, there was this scientist Dirk Nolf who was just amazing. He just had this sense. He was a phylogeny expert in otoliths, and there are whole manuals or whole atlases of otoliths that you can get to go through the different shapes, because they use this in diet studies and in archaeology. So if you're looking at bird diet studies, or you're looking at seal diet studies, you're looking at the scat, you're pulling out otoliths, and you can use that to look at the species, or gut contents for that matter. We use that. But I don't know why deep sea fish would have, if they were the same family, like a gadid or something, in a deep sea cod and a shallow sea cod. I don't know why, if deep sea cod would necessarily have different shape, size otolith. I think this is mostly kind of phylogenetic variation. Crystal morphology varies among larger classes. So we have vaterite in more ancestral fishes, and calcite in more ancestral fishes like coelacanths or they're vateritic. Sorry. And then yeah, you can use crystal type also to tell you something about phylogenetic groups.
All right, I have a question. Is the information that you found in terms of the survival size, the breeding size based on the salinity, the buffer, does that play into any fisheries management or catch limits per year? If it's a dry year versus a wet year, are the catch limits raised or lowered because of that?
Yeah, it really it doesn't go down to the environmental features, but more the end result, which is the juvenile abundance. The state tracks very carefully that juvenile abundance that they index each year with seine survey. And it turns out that that seine survey is that the juvenile abundance in Maryland tracks very well with the overall stock status of the Northwest Atlantic striped bass. It is our best indicator of it. Says it's really important. So right now, what you're going to read about in the paper is we've had our 7th bad year. And historically, that set off triggers for reduced harvest in a precautionary way. I think it will this time too.
All right. Steve also asks, have laser ablation techniques been used to analyze mercury in the rings? And if so, what does it tell us about the temporal variations and sources of mercury in fish?
Yeah, it has, but not a lot. And I think I've used laser ablation approaches and just ICP-MS approaches for looking at the really rare metals, the transient metals, the heavy metals in otoliths. And I've looked at eels because eels, they're very resident, so they can tell you something about maybe local sources of pollutants. And you definitely see some source things going on with the lead. Lead works really well for some reason in otoliths. Mercury is problematic in otoliths, maybe because of the speciation. With the metals, you're not looking at things that substitute into that nice calcium carbonate matrix. You're looking at things that glom onto the protein. And that's a harder kind of fractionation to understand. And particularly mercury with the speciation, it's pretty difficult. People have looked at it, but I think they're better markers of heavy metals like cadmium and lead that we can track better in otoliths.
All right. We have a question. What is your list of causes for the current drop-off in juvenile numbers?
Yay. Oh yeah, I'm on a big soapbox now. And I'm kind of, I wish I could maybe you guys can join my club, but yeah, I think it's blue catfish. It's absolutely blue catfish. They're our most abundant fish-eating fish, and they are just all over these nursery areas. A colleague of mine saw blue catfish following in the trail of spawning striped bass. Striped bass spawn up in the water column, and they opened up the blue catfish, and their guts were full of eggs. That's a learned behavior. These things know when striped bass are spawning. They know when juveniles are going to be concentrated in these areas. And I really think that this long-term depressed abundance, to me, looks like the blue catfish are controlling abundance. They're cropping this thing down until they get to a low abundance. I don't have direct proof, but it seems like a big smoking gun to me. That's what's different now than historic, at least in Maryland tributaries.
Eat more blue catfish, everybody.
Yeah.
Eat more blue
Yeah.
catfish.
Don't put them back. A lot of trophy fishermen, they want to catch them and put them back. No, don't do that. Eat them or use them for fertilizer or something. Get them out of the river.
Yeah. Yeah. Wow. All right. Any other questions? Alan put some information about the crystal alignment in the ears down there for you.
Oh, good. Found a reference.
And well, it sure has been nice seeing one of our heroes as well at the Natural History Society of Maryland, who started when he was a little boy with us. And that's where he got this spark and went off and did all these wonderful things and died way too young, because he would still be doing things. But look at his research is still out there being used and worked on. And we're working with David, or one of our Founders Day person who will be honoring next year in April for Founders Day. Our 98th birthday will be Romeo Mansuetti. And so he'll have him there to talk about it. We have some other things planned, so we'll keep you involved. But that was so exciting to see him and his work, and you utilizing it and kind of walking it forward as science always does. And I thank you for joining us, everybody. And I hope to see you next week when we learn about the manatees in the Chesapeake and why they're travel up. There's another migration story maybe for us with the manatees. And then, until then, be well, be curious, and we'll see you next week.